Welcome to the Protein Synthesis series. This video focuses on nucleic acid structure. At the end of this lecture, students should be able to describe the structure of nucleic acids and their monomer subunits, and to understand the nature of DNA being antiparallel and complementary. Nucleic acids are the biomolecules that make up DNA and RNA, which are the genetic material that is found in all cells. These blueprints store and transmit information from a cell nucleus and direct the cellular functions through protein expression. Just as protein macromolecules are made from long chains of amino acid polymer subunits, nucleic acids are made up of long chains of nucleotide polymer subunits. A nucleotide consists of a phosphate group that is covalently bonded to a 5-carbon pentose sugar molecule, which is covalently bonded to a nitrogenous base. Again, a nucleotide is a phosphate, a sugar, and a nitrogenous base monomer subunit. Nucleic acids have a negative charge on the end with the phosphate group. This means that it will always give up a hydrogen proton, or hydronium ion, when in solution. Nitrogenous bases come in 2 flavors, purine and pyrimidine. Pyrimidine have a single ring structure, while purines have a double ring structure. I try to remember that pyrimidine sounds primitive, and the more primitive would be simpler or come first. So pyrimidines are simple single ring structures. There are 5 nitrogenous bases. 3 of them are pyrimidine. We have cytosine, thymine, and uracil. The other 2 nitrogenous bases are purines. They are guanine and adenine. The nucleic acids do share many properties, but they do have differences as well. For example, RNA, or ribonucleic acid, is single-stranded, where DNA, or deoxyribonucleic acid, is double-stranded. RNA has a phosphate group bonded to a ribose sugar, which is bonded to a nitrogenous base. DNA, conversely, contains a phosphate group bonded to a deoxyribose sugar bonded to a nitrogenous base. Deoxy means without oxygen, so it would be a ribose missing 1 oxygen atom. Both nucleic acids contain 3 of the 5 nitrogenous bases, adenine, cytosine, and guanine. However, DNA contains a 4th base called thymine, whereas RNA's 4th base is called uracil. The nucleotides link together to form the backbone of the nucleic acid structure. Each phosphate bonds to an oxygen on the neighboring nucleotide's pentose sugar, which in turn binds to the next neighboring nucleotide's phosphate, with the pattern continuing to link the nucleotide monomers in that phosphate-sugar alternation. The bond between the sugars and the phosphates is known as a phosphodiester bond. The bonds are formed through dehydration synthesis reactions that remove a water by joining an OH group from the pentose sugar and an H+ proton associated with the phosphate group. Each nitrogenous base is complementary to another base. In both nucleic acids, cytosine always binds with guanine, so C equals G. This means when you see a C, there is always a G associated with it. And when you see a G, there is always a C associated with it. Conversely, in DNA, adenine always pairs with thymine. So anytime you see an A, it would associate with a T. And anytime you see a T, it would associate with an A. These complementary base pairs are associated with one another through hydrogen bonds. Hydrogen bonds occur between the electron hog oxygen, or nitrogen, and the electron apathetic hydrogen. In these bonds that hold the two strands of DNA together between the complementary base pairs, they look like rungs of a ladder moving up the middle of the two DNA strands. With RNA, the second pairing is between A adenine and U uracil. Chargaff found that if C and G must only bond together, then the number of C will always be equal to the number of G. As well, if A only binds with T, then the number of A will always equal the number of T. In light of this idea, we can understand that if there are 100 bases in a nucleic acid strand, and 22 are cytosine, then because of their complementarity and pairing, 22 must also be guanine. This gives us a total of 44 out of the 100 nucleotides. The other 56 nucleotides must be A and T. The only way that they could remain complementary would be to have one half of them as A and one half of them as T. This means that 28 amino acids are adenine and 28 are thymine. The same is true in RNA, only the number of A would equal the number of U. DNA from one of your cells stretched out in a linear structure would span 2 meters. That's the equivalent of 6 feet. DNA condenses in various ways to be able to fit within the cell's nucleus. First, it twists on itself into the double helical structure that we recognize, similar to the spiral staircase. It then coils around histone proteins. This is referred to as a nucleosome. The additional folding and condensing of DNA will be covered during our chromosomes lecture. DNA strands run in an anti-parallel fashion. This means that one strand runs from 5' to 3', and the complementary strand runs from 3' to 5'. The 5' end of the strand ends with a phosphate group, where the 3' end of the strand ends with the OH group on the pentose sugar. One complete turn of the DNA double helix is equal to 3.4 nanometers, while the size of one nucleotide is 0.34 nanometers. This means that there are 10 nucleotides in one complete turn of the helix. The spacing between turns in the helix is called the major groove. The smaller turn in the helix between the two full turns is called the minor groove. The minor groove is most easily identified by looking at the DNA double helix major groove and following directly through the helix to the opposite backbone. That is where the minor groove is located. Nucleic acids are DNA and RNA. RNA is single-stranded, uses the bases A, C, G, and U, and uses ribose sugar, where DNA is double-stranded, uses bases A, C, G, and T, and uses deoxyribose sugar. Each strand of DNA is made up of repeating monomer units called nucleotides. Each nucleotide contains a phosphate group bonded to a pentose sugar, bonded to a nitrogenous base. Each strand has a backbone formed by dehydration synthesis between phosphate and sugar groups of neighboring nucleotides, creating phosphodiester bonds. The backbones run anti-parallel to one another. One runs 5' to 3', while the other runs 3' to 5'. The nucleotide bases are complementary, so A only binds to T and C only binds to G. The only exception is in RNA, where thymine, T, is replaced by uracil, U, so A binds with U. Because the bases are complementary, there will always be an equal percentage of A and T, and always be an equal percentage of C and G. When combined, these four will total 100%. The complementary base pairs are held together by hydrogen bonds. As DNA twists into its helical structure, it also coils around histone proteins to begin to condense itself to fit inside the nucleus. Students should be able to contrast DNA and RNA. Students should be able to identify the components of a nucleotide. Students should be able to describe the complementarity and anti-parallel nature of DNA. Students should be able to explain what the backbone of DNA is composed of, and how it is formed. And students should be able to understand Chargaff's rule about nitrogenous bases.